So I think most of people here, we can start. So today we have another faculty candidate, Dr. Matt Breitenstein from Mayo Clinic. He is currently a research fellow in Division of Epidemiology, Mayo Clinic. Dr. Breitenstein received actually a long, when I look at your credentials, from bachelor degree, master degree in health service research, and a Ph.D. in human informatics, all from University of Minnesota. So you must have fun if you move here from Minnesota to Houston. Well, that's good. People, like President Obama said, we need change.
Exactly.
And your PhD is also in Institute of Health Informatics. The institute
Correct.
is very highly reputed, and my former colleague actually moved here for the director a few months ago, right?
Constantine.
Yeah, Constantine. I usually try to save time for the speakers because every time people always talk about a lot of exciting things. But the one thing I like to highlight here, Dr. Breitenstein has a R21 NCI National Cancer Institute fellowship, and this fellowship is specifically focusing on clinical cancer genetic epidemiology, and its very important thing is related to the pharmacogenomics and the pharmacological study. This is one of the most important areas right now in precision medicine, translational bioinformatics, and also one of the most important areas, the Center for Precision Health. That's the faculty position you're applying for. That's one of the most important areas we would like to develop. So welcome, and that's all your time. Yeah.
Thank you. So I'm going to give a talk today primarily focusing on metformin within the context of drug repurposing. We already went over my background a little bit. So it's cancer molecular epidemiology at Mayo Clinic is my current role. My research, while I'm a biomedical informatician, I think by training I consider myself, my research largely spans biomedical informatics on one end and molecular epidemiology on the other, with this kind of research focus on clinical pharmacology, which focused primarily in pharmacogenomics, but also within this cancer domain. So trying to kind of balance all these different perspectives, particularly the cancer research is no small component as well, but balancing all these different perspectives in my research. So like I said, I'll be primarily talking about metformin today, but I'm decided to wrap up my talk and add some work that I have ongoing in diffuse large B-cell lymphoma in a 5-agent therapeutic combination in RCHOP. And that's utilizing some existing epidemiology cohorts. So with that said, I'm kind of giving this talk from the perspective of both what is the biomedical informatics perspective and then also what's the epidemiology perspective. So if you have any burning questions as we're going along, feel free to ask it because other people in the room might be doing it. But a lot of what I try to do is balance kind of what we need to think about as informaticians, but really from an epidemiological perspective, the context and the details of what we do matters. So feel free to do that. So I'll start out kind of my talk landmarks. I'm going to talk about some metformin pharmacoepi work where I'm utilizing EHR data with some cancer registry data to study population level signal for metformin repurposing from diabetes to breast cancer. And then in the middle of the talk, I'll talk about some metformin pharmacogenomics and metabolomics informed pharmacogenomics work. And then in the last 15 minutes of the talk, I want to talk about the RCHOP pharmacogenomics. I'm not going to go into too many details on the disease for diffuse large B-cell lymphoma and the details of the combination therapy. I'm going to focus primarily on the metformin, but just keep in mind that with metformin, we're thinking about a single agent therapeutic exposure. With this other disease, we're thinking about 5-agent therapeutic exposure, and it only just amplifies a lot of the considerations that we've been thinking about with a single agent therapeutic exposure. So like I said, we're looking at metformin for potential breast cancer treatment repurposing, and we're utilizing EHR-based studies. So this is really applied informatics within pharmacoepi study design with some methodological enhancements to pharmacoepidemiology. So with metformin in type 2 diabetes, metformin is a first-line therapy. So with diet, you can use diet and exercise as well as a first-line intervention, but as a therapeutic perspective, all patients will be given metformin as the first-line therapy. And it has many different mechanisms for doing this, has many physiological mechanisms that it achieves this, and it's a very safe medication. When we think about it from a cancer perspective, we have these many pleiotropic mechanisms, with what we consider for diabetes, the beneficial effects as well that can have an impact on cancer, a beneficial effect. But we also have all these different biological bases for looking at metformin for repurposing. So this is one of those hypotheses that there's a lot of laboratory evidence that's accumulating as well, and there's a lot of interest from a population perspective as well. But we really want to see, can we see this signal? And so there's been a few studies that have been done looking at metformin repurposing for breast cancer treatment. Some have looked at all-cause mortality, some have looked at breast cancer-specific mortality, but really at the heart of the question is that since metformin is a first-line agent for diabetes, and anything beyond metformin monotherapy, so if you add combination therapy, there's other anti-diabetic medications, and eventually progress to insulin, there's a severity component to that as well. So the patients who are on different combinations of anti-diabetic therapies are actually going to be more severe and therefore have a higher risk of mortality events. But it's important to note that this is ongoing. There's ongoing clinical trials, but they tend to be focused within certain biological subtypes, and it's not clear what those results will be. So we have that initial, when we think about clinical response to metformin, and this is from the diabetic perspective, so how do we measure that phenotype? If patients respond initially to the therapy, so if their A1C goes down, their health outcomes improve, over time they'll become less responsive to that therapy, and their disease will simultaneously progress. So we have to think about that, and they add that combination therapy. There's this other subset of patients that while metformin is a very, very safe drug, that some of them just do not respond. They don't achieve glycemic reduction, so reduction in A1C, which is a clinical biomarker. And then there's about 5% that they just don't tolerate the medication. And most of those patients experience a minor GI discomfort, and they prefer to go on to some other therapeutic. And there's some rare major adverse events. We're not too worried about those today. But so it's kind of important to think about, if you want to look at the long-term exposure, a meaningful exposure from a pharmacoepi perspective for drug repurposing, we're thinking about the treatment severity. If we want to think about the pharmacogenomic implications, we really want to look at that initial response of the phenotype. And I'll kind of come back to this within that first two parts of the talk. So to emphasize this point further, we're really looking at, in the first part of this talk, what is the clinical trajectory which would be meaningful for drug repurposing in breast cancer. So where this standard pharmacoepidemiology approaches become very, very difficult is that it's a retrospective cohort where we can't intervene and give these medications to patients, so we have to make comparisons within existing subtypes. So if we're looking at a comparison, our exposure of interest is metformin for breast cancer, but what we can actually observe is metformin for patients with breast cancer who are currently on metformin monotherapy for type 2 diabetes. So we have this confounding by diabetes that it's always in there. And with that said, we can make comparisons, to breast cancer without type 2 diabetes. So, this would be what we make our hazard ratios or our odds ratio comparisons to, or, some other combination with metformin treatment for diabetes, no cancer. But what we actually need, cancer with metformin and type 2 diabetes, does not actually exist, and it is our most appropriate comparison. And there are other approaches that are needed to overcome this. And this is where we can look at this from an informatics perspective and try and utilize clinical trajectories, all this data we have available in the EHR to try and stage this trajectory. So really what we're looking at just conceptually is that we have our metformin exposure that impacts diabetes, and you can either think about it as modifying breast cancer, or it just kind of skews what we think about as the disease-specific mortality endpoint. And this comes back to where I was pointing out we had studies that were by all-cause mortality, some by breast cancer-specific mortality. So even with breast cancer-specific mortality outcomes, patients who are on metformin are more likely to have better outcomes just because the confounding disease state is lower risk of mortality. So that's one of the limitations we're trying to overcome. And within the pharmacoepidemiology literature, this is a big, big deal, the biases that are inherent in this. And we also have the sort of immortal time bias, time window bias, and time lag bias. But at the end of the day, all these different biases kind of give us what could be a beneficial effect for metformin that could potentially be inappropriate. So our approach that we proposed is trying to overcome the impact for some of these time-related biases, but also for the bias related to all-cause mortality, thinking about what we would experience with mortality outcomes. And does anyone have any quick questions? Did I lose anyone yet? No? So,
So in your previous graph, you mentioned cancer patient treated with metformin without the T2D.
Yep.
I was wondering which this kind of a clinical scenario is pretty popular, or it's very unusual in clinical treatment.
To have metformin and breast cancer?
Metformin is mainly for diabetes, not
Yep.
for cancer.
Yeah, and I guess that's a good point. So I was asked, is this kind of a common scenario where we're kind of looking at, we have two disease cross sections. So it's important to point out that both breast cancer it's a common cancer site, and diabetes is a common chronic condition. So you're looking at maybe about 5%, up to 5% of the population would experience both diseases within their lifetime. So we have it's more of a convenience question where we're able to look at within a few clinics based EHR to actually look at this perspective. So if you can kind of chop it down, but the cohort does get small. But so yeah, from a common disease perspective, this is kind of unique that we're able to do it. If you want to think about it from rare disease connections, you need to get bigger and bigger data and more representative cohorts to look at that. So, yeah. So, a few years back, I proposed a different methodology to do this, and this was extremely well received within the clinical pharmacology and therapeutics community, which has part of their core mission now is translational science. So I was not even a member of the society. They gave me their top training award. And what this is really trying to do is first account for the impact of mortality due to metformin exposure in diabetes first, and then try and offset that, apply that in a model where we're looking at breast cancer repurposing in kind of a novel way. So for the epidemiologists who might be in the room, we really like hazard ratios, and I was given feedback to include this as kind of a background estimate. So, with our cohort, if we did our standard approach where we try and adjust for all those time-related biases that we talked about and just using that standard approach, we'll come up with estimates for this cohort of where metformin is definitely protective, so it's 0.29 for the hazard ratio. And then for insulin, it's extremely detrimental. This is expected. When we think about kind of the depth of what we're going to talk about today, and these are, from an epidemiological perspective and clinically, these are nonsensical interpretations, but they're there for demonstration. But so if we stage out the diabetic component, you'll see that there is a risk, even for breast cancer-specific mortality for type 2 diabetes. And then you also have a split for breast cancer specific mortality. So later on, when you think about applying this novel methodology, you can go back and kind of do standard epidemiological comparisons, and you start to see estimates that, they're a little bit not as protective as before, but we're still, even with applying a very stringent offset, where we should really penalize away any potential effect if theoretically, if that's what's going on, if it's due purely to diabetes, we would expect this effect to vanish. But even with this stringent penalization, we're still seeing that there is a protective effect there. And for an alternative comparison, we actually see for insulin a detrimental effect. So this is really 3 different studies that are looking at this. One is how do you stage a diabetes trajectory? It's very common that with diabetes, if you can't achieve glycemic control, over time your A1C will go up. You need to increase your dosing of of metformin, and eventually you'll add in combination therapeutics, and then eventually insulin. So what's going on is your A1C measures are fluctuating over time. The clinical standard is to maintain A1C at below 7. So you're going to be altering these trajectories over time by clinical intervention. So we have to think about how do we actually stage diabetes correctly to mortality risk. 2 is kind of creating the offset using this methodology that we're proposing. And then 3 is thinking about developing standard pharmacoepidemiology models utilizing that. And I'll briefly go over each of these components today. So I utilized essentially 2 patient populations for these 3 studies, where we did a lot of our diabetes trajectory mining was in the Fairview healthcare system, which is based in the Minneapolis and St. Paul metropolitan area. It's also the University of Minnesota's healthcare system. And then as a comparison, we looked within our primary care population for type 2 diabetics at Mayo Clinic. And then our 3rd cohort would be a combination of primary care patients and referral patients for breast cancer at Mayo Clinic. So yeah, number 1 is our external cohort, and then 2 and 3 are internal cohorts. So the external cohort tends to be a little bit larger, and some of the characteristics are slightly different. And then our cohort we're looking at for the repurposing, so this is where we have breast cancer and type 2 diabetes. We can see that we have a patient population of about 860, and then we have for metformin monotherapy events, we have about 290 exposures. So part 1, we're looking at staging these, or identifying clinical components that represent this disease trajectory. Largely, I'm just going to summarize this in one slide. We think about, and this is kind of a goofy, how we abbreviate this, but we're really, in each of these models, we're adjusting for age. We need to think about A1C, because remember, A1C is our clinical measure that we're going to be modifying treatment. And the pharmacoepidemiology literature holds quite frequently that you need to adjust for A1C to represent disease severity. And so we wanted to see, do we actually need to include A1C? What else do we need to include? And so the complications and comorbidity, abbreviated by C and H, are, let's see here, are represented here. So we have our comorbidities, and then we have this list of complications that we include as additive effects. And then we look at our main drug exposure, so metformin and insulin, and the rest would be attributed to baseline. So the main comparison here, if we want to stage the diabetes trajectory, and these are predicting mortality of all-cause mortality events. So this would be, with a 1,000-fold bootstrap replication. So this would be kind of a standard data mining approach to just develop the best fit model. So we're looking at our fit for the predicting mortality events here. So if we think about metformin and insulin, we can achieve reasonable performance. If we look at the comorbidities and complications, we achieve reasonable performance as well. If we look at both, it gets better. If we look at A1C, it doesn't really add anything to that model. And if we look at A1C as a predictor alone, if we compared age and then adjusting for even the complications or what have we here, the performance starts going down when compared to metformin and insulin. So this, for our study, we wanted to stage this trajectory. It also has some implications if you don't need that clinical biomarker. You can think about utilizing this approach from a larger claims repository, where you might be able to only see that this test was run. You may not be able to see what the actual value was. So it kind of has creates some multiple opportunities. But so for this purpose, we're going to think about modeling all-cause mortality with metformin and insulin from a very kind of simplistic perspective, so that we can clearly show what our comparisons were. So part 2 is where we actually have this methodology. And it's important to note that you can do this with either Cox models or logistic regression. So if you want to have a time to event modeling, or if you just wanted to do dichotomous outcome, you can do that as well. So really what we're doing here is, now that we've identified the components necessary to predict all-cause mortality, we're gonna create a model where we fit that, and then we're gonna take the beta coefficients out of that model from a stratified analysis, and then apply that into the breast cancer specific mortality model so that we can make these comparisons. So when we think about training it, we have our model components here. And then we see that, if we just look at this alone, the breast cancer cohort doesn't reasonably predict this, because this is where it's contained, where it has both the diabetes and breast cancer effects. If we think about creating the second part, where we create this offset using our Fairview cohort, we see into our Mayo Clinic breast cancer cohort, we see that we improve model performance, notably using these two different perspectives. So we see an increased model performance, but we're also, from an epidemiological perspective, we're accounting from that clinical trajectory in a more robust way. And there's many, many things that we could talk about, nuances of how do we actually fit these different things, what are the implications of it, and I'm happy to kind of talk offline if we want to. And then in part 3, now we think about adding in the hazard ratios and applying it in an epidemiological perspective. And so these are hazard ratios, not odds ratios. And this is that same slide that we saw before, where we look at, if we think about it from targeted therapy perspective for breast cancer, what they're actually prescribed, and then also what we see from receptor status from clinical notes, we see similar hazard ratios. So this is one of the studies I'm working on writing up right now. And I would actually like to include a second cohort to replicate these findings on. And so from thinking about the receptor status phenotype, so this is what the comparisons were based on, this treatment perspective, receptor status perspective. You can even see that the informatics phenotyping perspectives matter quite a bit in these methodologies. So when we think about these drug repurposing studies, we have both informatics considerations that we need to account for and epidemiological considerations, and it's important to kind of balance both of those as we proceed forward. So the strength of this methodology is that we're enabling an alternative comparison that did not exist, but it's also important to note that it doesn't actually exist. We're creating sort of a pseudo-counterfactual to compare. It's interchangeable for both Cox and hazard regression, or Cox and logistic regression. And you can work on the complexity of that clinical trajectory as needed, which is important. It also gives us the opportunity to utilize an external, larger, potentially more balanced cohort. And what we saw is that if we don't need to include A1C measures, it's actually possible to utilize something like an insurance claims repository, where we have 10 to 100 times the number of patients to try and establish that clinical trajectory. So this doesn't replace the prospective clinical trial, but it does give us this alternative comparison. And like I said, it's important to balance both informatics and epidemiology perspectives, because there's potential to introduce bias, great potential to introduce bias if you're not careful with each of these steps. You can introduce a whole set. And it's a minor consideration, but this is, the clinical data requirements are beyond a traditional cancer registry. For a lot of folks, this doesn't matter because we have to, for like SEER, we have to collect clinical repositories and all the meaningful clinical data points, but we actually do need to utilize EHR data and potentially insurance data to kind of augment these clinical data repositories to do these approaches. So this brings us back to the previous slide, which I call our inflection point. So we think about, we've kind of covered if patients respond to this therapy, kind of what happens over time. We have this other, about a third of the patients that, refractory is kind of a bold statement, but really they just don't, they're not responding to treatment ideally, and there's likely to be some sort of pharmacogenomic question. And this starts to paint the broader picture of, when you're thinking about all these pharmacoepi studies, what are our actual clinical phenotypes that we're looking at for repurposing? We can think about that we have clinical implications that we're capturing, but we're also thinking that some of these clinical implications have pharmacogenomic implications, others don't. But so here we're focusing primarily on pharmacogenomic implications with this A1C response. And so it kind of paints this need that there's going to be opportunity to enhance the phenotyping we have that are kind of spanning diseases within the EHR. But there's also perspectives, opportunity from a molecular perspective to think about biomarker development, where we utilize clinical samples paired with electronic health record data to drive discovery, but then also kind of validate precision medicine knowledge or develop precision medicine knowledge. So part 2, I'm going to talk about some metabolomics and pharmacogenomics work. All right, got a few minutes here. And so we utilized, in the first part, we're utilizing A1C as a clinical phenotype. And then towards the end, I'm presenting some of my ongoing work utilizing metabolomics as an endpoint to inform pharmacogenomic inquiry. So just a really quick crash course on pharmacogenomics. We kind of have 2 considerations. We have kinetics, pharmacokinetics, which impacts bioavailability. So you either transform the drug into an active component or you need to move that drug to the correct organ site. And so there can be variation within pharmacokinetics that can just impact, is the drug available, right, to that organ site. Another perspective is the pharmacodynamics, which is how is it interacting with different physiological processes. And I'm not going to go into too many nuances today about kind of defining those, but it's kind of important to note that there are actually 2 sets of complications, or 2 considerations there. With metformin, we really understand the pharmacokinetics well. We understand how it gets pushed around. It's not biotransformed, but we really don't understand how it works. We have all these disparate mechanisms that we need to put together. So when we think about drug repurposing, we can look at it from this clinical perspective to see that, yeah, it looks like there might be a signal there, but really, what at the heart of it, what is it actually doing in a human population? So these are from so this is from 2 of my mentors put this together. This is what we know the state of pharmacokinetics. It's absorbed, and then eventually you excrete the whole product. And then with pharmacodynamics, we have all these disparate mechanisms as we kind of saw before on one of the opening slides. So one of the pharmacogenomic studies where we looked at utilizing a clinical endpoint to inform pharmacogenomic inquiry, we really saw that we utilized glycemic response, but it was not a very robust phenotype to drive discovery. We did get some associations, but they're nothing too great. We got one association within this PRK-AB2. So at this diagram, it's basically the beta-2 subcomplex of this mechanism this enzyme complex that sits at the center of really what's known about the kind of the direct pharmacodynamic effects of metformin. So we picked up a signal there in the beta-2 subcomplex, beta-2 subcomplex, which is great. But really, it doesn't give, and there were no genome-wide association hits for this study as well. We did look at a GWAS hit, and there's not anything terrible novel that came out of this, but it's not clear that that represents what we're looking for. So a lot of times the clinical phenotype that we have available, existing clinical phenotypes, may not actually represent treatment response for the actual question that we're trying to answer. Particularly the biomarkers vary in biological significance. So while they may have important implications biologically based for clinical management, they may not actually speak to biologically what's changing, how are the patients actually responding to the drug. So metabolomics offers one solution to try and approach this. So with metabolomics, there's kind of 2 broad approaches to identify the metabolites. We can do a naive approach, which for many of you that took organic chemistry back in the day, you would have seen you have to identify all those peaks and try and figure out what the compounds means. And there's a lot of ongoing work within NIH resources, particularly within NCI, to develop more and more metabolite standards. But it's important to note that there are a lot of metabolites that you can identify those peaks and you just won't know what they are. With a panel approach, it's well determined what these metabolites are, and you can actually quantitative measure the concentration of the metabolite in your sample. So what we chose, and it's also biologically hypothesis-driven panels. So we chose an amine panel, which is really focused on some of the mTOR pathways, which would be kind of on the far right of one of the opening slides of this pathway, which is involved in protein synthesis. So we used the tandem mass spec liquid chromatography platform, and there are others available, and they all do slightly different things, but it's just kind of important to see that there's all these different approaches. So kind of the central dogma of metabolomics to inform pharmacogenomics is we're utilizing biological activity biomarkers. So you have to first make that association and then elucidate these pharmacogenomics. And primarily when we look at metabolomics, when we can control for drug concentration, so we can measure the actual drug concentration, we're controlling for many of the pharmacokinetic determinants, so which determine how much drug gets pushed around to different sites. So we're really looking at kind of the novel determinants of what's the disease and how does this drug, what is it actually doing in the context of normal tissue and then also within diseases. So within our biobank cohort, which is utilizing as kind of the forefront for the precision medicine cohort now that'll be based at Mayo Clinic. This is utilizing the plasma samples that were after the initial testing and stuff. This is where we accumulated our biobank of about 50,000 patients, and we utilized plasma samples for research. So there's a few different things going on here, but we're making case and controls comparisons for metformin and type 2 diabetes. Again, we have that confounding by diabetes. And we're looking at 42 metabolites. And we age and gender match the cases in control, and we can adjust by batch and BMI. So it's evenly matched, 276, 276. For reference, these are the metabolites that we looked at. And what we see is that there's a correlation between the dose and the concentration that we observed, some of our cohort characteristics, and then looking at some of our covariates as well. So this is what impact of BMI on daily dose, and then also concentration. So as BMI increases, the actual bioavailability of metformin slightly decreases. But we adjust for these covariates in our GWAS analysis. So we're looking at utilizing some existing studies to help us frame what of these metabolites is a diabetes association, what might be relevant to metformin alone. And what we utilize is a Framingham study as kind of our baseline comparison, and they very clearly established metabolite expression profiles for diabetics versus healthy normal control, and they're not exposed to medications. So we focused our analysis on 14 metabolites that are not statistically or marginally significantly associated with diabetes, and we ran our analysis to see which of those changed with metformin exposure. And we also had as a replication cohort a human variation lymphocyte cell line panel, which is the basis for a lot of the research coming out of the Pharmacogenomics Research Network at Mayo Clinic. So we identified 3 metabolites that were statistically significant. 2 of them decreased, 1 of them increased. And then we had cell line replication for directionality. Due to the effect size, we didn't see a statistical significance. We were primarily focused on the directionality. We had 1 metabolite that was very highly statistically significant in the human population, but the LCL model was statistically significant in the opposite direction, which was interesting. And then these 2 metabolites are actually within a reaction of each other. So our ongoing work is going to focus on if there's some sort of, if we're looking at different concentration ratios between those 2 metabolites. So some of our collaborators and competitors were also looking at metformin metabolomics from a much smaller cohort, 33, prospectively. And they collected these samples at UCSF. And this was actually published, I think, last week, Wednesday, it came through. And we've known this for a few months now that they're seeing that citrulline decreases. So they're replicating that finding, and that glutamic acid is increasing as well. And this is in an African American cohort. And so this is not my research, but it's important to note that we do actually see these metabolites replicating from our human plasma samples that we did in the biobank, collected with type 2 diabetes and metformin, replicating in a healthy, normal cohort, prospectively captured with controlled diet and exercise. So I'll focus on some of the citrulline, what we can do with this. So what we see is, one, we're seeing a dose-response relationship between citrulline. So as a metformin concentration increases, you're seeing citrulline decrease. You see that with daily dose as well. And citrulline is interesting in that it's implicating the urea cycle. So we have this sort of biomarker here of what's going on, but it fits within these larger reactions and within this larger pathway. So when we look at utilizing just the citrulline association, which decreased, to inform pharmacogenomics, we see one hit, or a few different hits. None of them are genome-wide significant, but this tends to be what we see when there's potentially some really functionally relevant things. So this may not be directly these genes involved, it may be something else. And then I'll be presenting these findings in depth, the pharmacogenomics findings for the 3 metabolites, next week at the Metabolomics Society meeting in Dublin. And for reference, we have the locozoom for this gene as well. But as we can see, just to kind of briefly wrap this up, that it's quite complex to think about drug repurposing within a single disease, single drug exposure model, and also to quantify what does that potentially do. So it really kind of hammers the point that thinking about precision medicine targets, with metformin, where we know a lot about this drug, and we still really don't understand the pharmacodynamics. So if we want to think about repurposing the drug or repurposing the target, there's many different approaches we can take utilizing EHRs to look at that, but then also things like the precision medicine cohort, opportunities for collaboration such as here, with MD Anderson and some of the other clinics, where we can prospectively, or we can look at collecting some biological specimens that relate to some sort of disease trajectory. So we can utilize methodological advancements. We can utilize biomedical methodological advancements to drive robust epidemiological inquiry. But we do need to think about incorporating study design, minimizing error at both the biomedical informatics phases and the epidemiological study design and interpretation. And this is also potentially interesting utilization of the precision medicine cohort, that if we do want to look back and try and do metabolomics to inform a pharmacogenomics association, primarily if we've established this trajectory in a larger EHR cohort, we can potentially utilize that there. So I'll kind of wrap things up, and I think we're right on time here. So do this pretty quickly. So now we're looking at an immune system cancer where we have to primarily rely on therapeutics. Whereas breast cancer, you can take out the cancer site if you don't respond. So precision medicine is very important within this cohort, and so I'm not going to go into too many details about this, but it's a 5-agent therapeutic. One of those agents is a biologic. And so the RCHOP is specified by brand name of the drugs. But so approximately R is our biologic, C, cyclophosphamide, is our chemotherapy agent, which by quantity is the major drug product. And then we have these different medications as well that together they have a synergistic effect. And then DLBCL is non-Hodgkin's lymphoma with a disease subtype of diffuse large B-cell lymphoma. And so this is some of my work that's coming directly out of the NCI-sponsored postdoc, where this is kind of bread and butter molecular epidemiology with a pharmacogenomics twist. Our pharmacogenomic knowledge base of combination therapies is extremely limited. So what we've seen, what we know about single agent therapies, but when we combine them, we may not know the pharmacodynamic determinants at all. And we also don't think about, multiple diseases, which is a focus of my ongoing research. So we want to look at pharmacogenomic knowledge for combination therapy. We're using clinical drug response within an epidemiological base data set. And refractory in this case is obtained by a standard clinical assessment of this Hans algorithm, and it's the closest clinical phenotype to drug response that we have available. And we have, here we have 2 independent cohorts. One's a US cohort at Mayo Clinic, and we have about 424 patients. We have about 10% of patients are refractory to treatment, so they don't respond to the therapy. And then we have our independent cohort in France, part of the Interlymph cohort consortia, which the meeting is actually going on right now. So I'll be flying home, and then I have to present at the Interlymph meeting at 9 in the morning. So, because I got to get my DLBCL hat on right after this. But we have 2 independent cohorts, and this is really what we can, a really powerful statement on what we can do with epidemiological data sets to think about precision medicine as well. And this is where we pooled those 2 cohorts together with a meta-analysis. So it's not a pooled analysis, it's a meta-analysis. And we have 3 signals, one of which is genome-wide significant. Within the Mayo Clinic cohort, this association is genome-wide significant, but these signals both replicate as independently, and then these are the signals as a meta-analysis. So we have the agnostic association of MAN1A2, and there's a little bit that's known about it, what it does in the Golgi. And then we have part 2, which is a Parkinson's gene that we got to hit on. And so we're actually seeing a gene that's linked to a different disease association that's showing up in this sort of where we break it with a drug exposure. And then we also have, utilizing some existing pharmacogenomic knowledge of cyclophosphamide, we see what we can gather with a candidate approach as well. So we see with MAN1A2, we have a fairly wide signal across the gene, all statistically correlated with the SNP that we identified as our top SNP. So we're looking at going back and doing a 3rd set of replication for technical, and then also within our cohort to see if it replicates. And then also doing some, have collaborators that'll be doing some functional characterization soon. And we see that the tissue that this gene is expressed in is actually very disease relevant. And if from a kinetic perspective, it could be relevant as well. But this is where we're utilizing some existing bioinformatics resources to characterize if it's potentially a meaningful hit or not. So we see in both B-cell lymphocytes, DLBCL is a B-cell-based cancer. We see it in whole blood samples as well. For part 2, we see a very focused signal coming within one LD block. And we see some very limited exposure or expression in B-cell lymphocytes, so we see it in other organ sites. So it's not immediately, neither of these associations, it's clear what they mean, but they're potentially useful implications to help us form future research. But it does form in the pituitary, which that tissue can form lymphomas as well. It actually forms in the B cells, but that's the surrounding tissue. For our candidate study, replicating our previous work on the Mayo Clinic cohort, we saw that this one association, which was in that genome-wide association that I showed you, the Manhattan plot with the genome-wide associations, we see that one of them replicates this ALDH5A1. This CYP2B6, which we previously characterized, did not. And the C in cyclophosphamide, when we look at liver, so this is where the biotransformation of this drug occurs. We had previously identified this signal that didn't replicate, but it's important to note that cyclophosphamide is actually biotransformed in the liver by this series of biotransforming enzymes into this active product that it sits in equilibrium here, and then it's sent outward, diffused outward to the cancer site. So it's important to note that it has to be biotransformed, so cyclophosphamide's a prodrug, and then the implications we have to think about within the cancer cell. And so this was our other hit that we had previously that replicated. And what we see here is that this activated product, that's bioactivated in the liver, is now actually deactivated, excreted out into this product in the cancer site. So if you have variation potentially within this gene, you could be impacting the expression of this product that deactivates your activated drug product. This is kind of an interesting precision medicine question. And when we look at the signal itself, we see it here. This is ALDH5A1. What we see is that the near neighbor is actually where the signal is coming from. And so just as a very kind of surface level inquiry, we're interested to see if this gene here, which is DCDC2, or the SNP is potentially functionally related to this gene. And again, this is using that GTEx project. And we see that while ALDH5A1, so it's a biotransforming enzyme, we would expect biotransformation, to be expressed in the liver, but we also see it expressed in B-cell lymphocytes as well. Whereas with DCDC2, we see no relevant expression in either liver or B-cell lymphocytes. So it's likely to be impacting expression of the ALDH5A1 gene. So you can do this thing called Eqtl Analysis, and this starts getting into what you can do with bioinformatics characterization to see if the SNP, which is this signal here, just as standalone, is associated with increased change within these tissue types for this gene, the ALDH5A1 of interest. And we see that it does significantly impact ALDH5A1 in relevant tissue types. So, again, I included this work not necessarily for its biomedical, for informatics novelty, but to kind of show that what we can do with utilizing existing epidemiology cohorts, but then also to demonstrate when we think about 5-agent combination therapies beyond just a very simple exposure, it gets to be quite a complex. And you do have to have kind of a balance of existing cohorts and then what we can do with EHR mining. We have some findings that might be determinants of treatment response. And this also contributes to an evolving knowledge of different subtypes of diffuse large B-cell lymphoma as well, which all lymphomas, surgical interventions are extremely, extremely rare. So within this cancer type, you really need to rely on a precise therapeutic, that your patients will respond to. So even if it is 10% treatment non-response, it's very important because these patients have a really, really high risk of experiencing a mortality event very soon. So kind of where does this all fit within precision medicine, precision health? We see that advances in informatics innovation drives advances in discovery of novel biology, and also, primarily here, identification of treatment personalization opportunities. And we do this through innovative integration of existing knowledge, development of novel methodologies, epidemiological rigorous study design, innovative scientific inquiry, and of course you have to have collaboration across different areas of expertise as well. So I've talked in depth to many different people today about kind of future directions for my research. Some of the things that I'd kind of want to highlight here is that there's potential to further develop biomarkers to inform genomic studies as well. This can have different areas of work. Also, within precision medicine, some informatics methodologies I want to work on is how do we think about polypharmacy? So it would be multiple drugs, multiple diseases, and that account for different aspects of severity, what's known about the targeted aspects of the therapeutics, and then the actual disease etiology. So one of my main, I think, 10-year goals is to be able to account for these kind of different combinations of things with multiple drugs, multiple diseases, and how can you predict if a patient, that therapeutic is appropriate for that patient. And then also looking at this area that spans the known clinical phenotypes at present. So, our example, it's unlikely, we're not proposing that breast cancer and diabetes are related, but you could also potentially look at phenotypes between them to see if there's something that might link them together. So there's, and this could be adverse events, minor or major, or just even anecdotal findings that you find from what we talked about today with someone, even looking at like Twitter feeds or something where patient takes this drug for this disease and they also have this other condition and they attribute it to the drug, but it may actually be something they're just perturbating some aspect of this other disease as well. So I think there's a few different directions we can go from a biomedical informatics perspective as well to help inform this research. I want to acknowledge kind of my key three mentors in this work, Dick Winchellbaum in the Center for Precision Medicine at Mayo Clinic, Li Wei Wang, they're both in the experimental therapeutics group, and then my epidemiology mentor at present, which is Jim Serhan, and he runs these interlymph cohorts that I was working on. And then also I have some bioinformatics, metabolomics, and EHR medical informatics collaborators as well. My funding primarily comes, at present, my salary is completely supported by this R25 in molecular epidemiology. Previously, my dissertation work was funded by the Pharmacogenomics Research Network, and then this other support helps fund some of the projects specifically. So with that, I'll end the talk, and I want to thank you for attending. And if you have any questions, please feel free to ask.
Let me ask you a question first. So in your second part, you mentioned metabolites association with diabetes. I recall you have three kind of metabolites not associated, not studied,
Yep.
and found to be statistically associated with diabetes.
Yep.
So you follow up, as a follow-up analysis, you use those are not associated with diabetes to further.
Yep.
Yeah, this one.
Yep.
So why not look at the second tier, those metabolites associated with people disease, and then see which kind of it is contribute to maybe associated with the metformin treatment, or not because of the metformin.
Yeah. So like pharmacometric analysis. Yeah, I definitely want to proceed forward with that. So yeah, that's actually an active. That's one of my next planned projects actually, because there's actually some really robust information we can gather from this. The study we want to gather, just for a clean publication strategy, is that we have this thing that's not associated with diabetes. But yeah, it's some really interesting things that we can gather, and we do have the drug concentration levels as well that are measured. So it's a really powerful cohort, and it's linked up to the EHR. So yeah, there's many different things we could do with this.
From clinical perspective, for these two kinds of follow-up analysis, which one you consider is more important?
For clinical perspective?
Related to these, whatever pharmacological stuff.
Yeah, so I mean, if you think about it, and you briefly alluded to it on one slide where it kind of broke down the different biomarkers, right? So we have this sort of level, it's like levels of biomarkers. Some where it's just, one can be prognostic, so the biomarker would be independent of therapeutics. So you would be able to predict likelihood of survival, severity of the patient, independent of treatment. So pre and post drug exposure, it'll be the same, but it'll predict it. And then you have these sort of, on the other end, you have clinical biomarkers, which are validated to have a clinical endpoint. You start going back, and you have biomarkers that are associated with the clinical phenotype that can be proxies for it. And so there's some powerful things you can do with that. Here we're looking at discovery biomarkers where it's measuring biological activity. We can relate it to specific reactions within specific pathways. So it's measuring biological activity. In terms of clinical relevance at present, this is where I think we can utilize these cohorts to drive the actual knowledge base of precision medicine, whereas we can utilize, I think, either proxy biomarkers or clinical biomarkers to help inform what might be more kind of a low-hanging fruit in terms of actionable clinically actionable precision medicine as well.
So I'm curious about in the Archon project.
Yeah.
How is it decided that those 5 drug combinations?
So one, it's based on the DLBCL disease. So it's kind of a specific, for that specific disease. So this is just, it's kind of the standard of care for those patients. The caveat is there's a lot of active research going on in biologics and utilizing the immune system to harness cancer. So we actually see the R is changing, but this CHOP regimen is kind of a very well-established standard of care that's been around, and it's kind of got all these different mechanisms that overlap. But
So all 5 agents are used at the same time?
Yes, so they're delivered at the same time intravenously. Yeah.
This is quite new to me. Overall, in pharmacological studies
Yeah.
or the application to clinical care, is this not the level of the combination, like 5 drugs together,
Yeah.
will become trained, or it's already kind of like very popular in the pharmacological?
Well, yeah. So is this common to have five therapeutics, or is it going to become more common? And I think the reality is we treat patients as we think about research as if we are single agently treating patients and we have one disease we're treating. The reality is we're not. So I think there's that where it's you can kind of think about that. But I think even with breast cancer we see we have our neoadjuvant treatment, and then now we're seeing extended adjuvant treatment. So that's you can give chemotherapy, but you're also giving anti-estrogen medication for 5 to 10 years following treatment. So there's implications from a slightly different perspective of multiple agent combination therapy. But yes, I think it's going to become increasingly more common. There's a large movement within the pharmacology community to obviously have more precise targets. The problem is they have to be working on average for all patients. So it's a really interesting arena because policy is shifting from pharmacology. We're getting more knowledge about precision medicine. And I think on the one hand, we're going to have more combinations of therapies that are kind of custom designed for patients. In terms of having a lot of these kind of like blockbuster combination therapies, I suspect that's going to decrease over time, but it is going to the actual number of kind of curated combination therapies where we're looking at specific drug, cancer targets across cancer sites. I think that's going to increase. So.
Okay, I may ask you one more question. So I really like your work in Metformin and diabetes. This is a very important area in pharmacological study as well as epidemiological study.
Mm-hmm.
So, there's one kind of study is those cancer patients, those kind of diabetic patients treated with metformin are less likely to develop cancer. So, like in your study, have you thought about that, or potentially, what do you think about that?
So the question is, so it's known that metformin it decreases your risk of developing breast cancer, and
overall cancer.
yeah. So that's actually one of those time kind of it broadly included one of those time-related biases that you need to account for the total exposure of metformin. And theoretically, this could actually modify. So if you're taking metformin for 2 years before breast cancer develops, it could apply selective pressures so that the breast cancer etiology could be slightly different, or it could be more severe, less severe, or something like that. So yeah, it's an important consideration to account for. It's actually one that we can't really account for in our cohort because the majority of our breast cancer patients are referral patients in the upper Midwest. So we have a really good job of picking up if they're on metformin at the time of breast cancer treatment, but we can't ascertain how long they've been taking the drug. So yeah, but it's an interesting caveat with this work, and it's a really difficult area to try and propose a repurposing study. So yeah.
Okay, so any other questions? Otherwise, please join me. Thanks for today.
Thank you. Yeah.
So somehow the data you can bring here, also the new data here.
Yeah.
It will be very helpful for your future research.
Yeah, definitely. And I've worked out